Hey everybody, we're just going to start out by explaining a little bit. We had a little bit of trouble meeting with our group 7. So we're group 7. Some of the other guys weren't able to come, so we're still figuring out how to coordinate our schedules. So Jordan and I decided to do it ourselves. We met with Kelton and Joseph for a little bit, but they're finishing up the worksheet right now. But we have stuff going on a little bit, so we are making the video, and then they're going to submit a separate video. Hope it's okay, Sister Mayo. We love you. Please don't deduct points. I'm gonna delete that. Writing on the board is gonna be a little bit difficult because we realize it's gonna be backwards. So I don't know if that's gonna be a help to do. But basically, we're gonna go through each question and talk about how to do that. So first, let's move this table so I can screen it. The first question is talking about epigenetic inheritance and Angelman syndrome.
Oh, okay, yeah.
What did they say?
So come in when you're done.
Okay. An Angelman syndrome, which displays genomic imprinting. So epigenetic inheritance is when, the simple explanation is we just ignore the male allele or the male gene that comes when you do a cross. So we know, so the question is saying when you cross a heterozygous male and a heterozygous female, what happens? So you do a normal cross and you get a homozygous dominant for one, 2 heterozygous, and then a homozygous recessive for another. And a good way to look at it is if this is the male right here, we can just take and say, okay, this one is not reliable, doesn't matter. This one doesn't matter. And then these 2 don't matter because those are the male alleles. So we would just look at what allele is displaying it. So we would get 50% of the offspring would be normal because a dominant allele is normal. And then 50% would have the Angelman syndrome. To do this, you do the exact same for B, except for it would be a little a for the female, I believe. Yes. So you're going to get A. And you just do the same thing. You cross that. And since the female is homozygous recessive for the gene, she's going to always give a allele that causes Angelman syndrome. And since the father's allele doesn't matter and you don't take it into account, every single one of them will have Angelman syndrome. Part C is a little bit more difficult. It talks about the Barr bodies. So how we understand it is that when you have 2 of the XIC parts on the X chromosome, that makes a Barr body. So this one says if a triple X, so she has 3 X's, has in a woman, and then she only has one of X, won't get any Barr bodies because you need 2 to make a Barr body. And then for D, it's a little bit more difficult. It says suppose that you isolate a cell from a female cat with 2 X chromosomes. However, the T6 gene on the X chromosome are non-functional, while the XIST genes on each X chromosome are functional. How many Barr bodies are there expected to be found in the cell? So you have a normal cat that's got 2 X chromosomes, and this would make it where 2 of them they're gonna get 2 Barr bodies because the XIST will bind to the T6 and inactivate, which causes the production of a Barr body on each chromosome.
Okay, so number 2 is fill in the blanks. We found these throughout the notes and just different sources. A asks for an example of a holoandric gene, and from the reading we found that SRY is an example of that. B asks for an example of a gene found on both the X and the Y chromosomes, and that is MIC2. Missed that on the quiz. It is found on both the X and Y chromosomes. So C asks for a cross that is made between IGF2 male mouse and an IGF2 female mouse.
That's an example of when you don't use the males.
Okay. Yeah. So it asks, what are the phenotypes of the offspring? And so looking if we don't use the males, we'll get 50% normal and 50% dwarf for the phenotypes. And that's just through a cross. And like we done the other one, you cross out the male genotype, and that would give us 50% normal, 50% dwarf. And for D, in the sex influence pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other sex. And so that, like I read off, it's sex influenced. And that's like, for the example, the goats. The beard allele for males is dominant, but it's recessive for females. For E, it says genomic imprinting. That's one of the blanks. Is an epigenetic process that occurs during gamete formation, while dosage compensation is an epigenic process that occurs during embryogenesis. For F, one of the topics discussed in lesson 2 involves the formation of a double-stranded RNA molecule. What two molecules form the two halves of these double-stranded DNA? And through the reading, through looking through the X center, we find that the T6 RNA and the XIST RNA are those two that form the double-stranded RNA molecule. And G says when reciprocal crosses give identical results, the allele that controls the trait in question are located on autosomal chromosomes. When the reciprocal crosses gives different results, the allele that controls the trait in question are located on the X chromosomes. And so that through doing different recessive reciprocal crosses, what we do is we take the homozygous female dominant with a recessive male, and we'll get a certain genotype, and then we take the homozygous recessive female with the dominant male, and then if the phenotypes are different, then we see that that is X-linked, and if they are the same, then we see that that is autosomal. Okay, so number 3.
All right, we're gonna speed this up because we're only supposed to make 15-minute videos. So
What do we have?
I don't know, it doesn't say. A woman's father has OTD in an X-linked recessive disorder. So saying that he's X-linked recessive, so he has it, so he has the recessive and a Y. And a woman is homozygous and is unaffected. So if she's unaffected and homozygous, she has to be X dominant. So basically that's what the answer for A is, because you just have to figure out what the possibilities are. B, this asks if the woman that is the daughter of the two parents in A, so she's going to be heterozygous because she's going to get a little a. We used A for the superscript. Little a from her father, and then a big A from her mother, so she can be heterozygous. And it says with a man who does not have OTD, so he is X big A Y. So if that happens, what is the percentage? What is the chance of getting their child? It's going to be a 50% chance, because if she has X A big A and then X little a, this is going to be the 50% chance because he's going to always give both Y's. So that works. And then for C, it says if they're going to have a, what type of genotype does the male have to be to produce a daughter with OTD? The only way to do that would make it where the man has OTD, just like the woman's father, because he will give an X chromosome to the daughter. And then for D, it says consider the cross described in part C of this question. What are the expected genotypes and phenotypes of the offspring? So we'll just do that cross really fast. If he has OTD, so he's X, A, Y, X, A, and then X, A. We're gonna get X, A, X, little a, X, big A, Y, X, little a, X, little a, X, little a, Y. So basically, we're gonna get 2 males and 2 females, and each one of them will have, won't have the disease and one will. And so it's gonna be 25% female with disease, 25% male with disease, 25% female without disease, 25% male without disease. And then number 4.
Okay, so number 4 is an example of sex influence traits. it's talking about goats. It says that the presence of a beard in some species of goats is determined by an autosomal gene that is dominant in males and recessive in females. And so it says that suppose that a cross produces 3 bearded goats and 1 no beard for the phenotype ratio among male offspring. So this is just male. And it says, what are the genotypes and phenotypes of the parents? So we see that both parents are going to be heterozygous, because if we look at the male, it means that if it's going to have a beard, it has to have at least 1 dominant allele in it. And so if we cross these, we'll get 2 heterozygous, 1 homozygous dominant, 1 homozygous recessive. And so these 3 all right here are going to all have a beard, and this one will not. So that gives us the 3-to-1 ratio for A. So both parents are going to be heterozygous, and since the heterozygous for the male is large A, little a, then it'll be a bearded. That's its phenotype, and the female is going to be have no beard, because the A dominant A is not a beard.
It has to be heterozygous.
Yeah.
Or homozygous.
Homozygous recessive in order to have a beard. So for B, it says suppose that a cross produces a 1 beard, 3 no beard phenotype ratio among female. What are the genotypes and phenotypes of the parents? So this is going to be the same as A. So if we were to look at, so for a female to have a beard, she would have to be homozygous recessive. And so we see that these all have a large A in it, which means that that would not have a beard for a female, but this one would. So it's the same cross with the parents. They're both going to be heterozygous, but instead for females, it's going to give us the 1-to-3 ratio. And for the parents, it's the same. The father will be bearded and the female will not be bearded. For C, suppose a cross between a male with a beard and a female without a beard produces a 1 beard to 1 no beard phenotype ratio among the female offspring. What are the genotypes of the parents? So it's going to be a 2-to-2 ratio. And the genotypes that we find are going to be BB for the male, which is going to be the genotype with bearded. And for the female, we had a heterozygous BB. So this gives us BB, B, 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 little b, and B, little b. And so, and this is going to give us a 2-to-2 ratio for those females.
Yeah.
Yeah. So for females.
Because the big B,
Yeah.
It has to have both. It has to be homozygous for the dominant, or they call it the recessive, but we're going to say that the big B in female is the recessive trait.
Yeah. And then for D, it says suppose a male and female, both which lack beard, so the phenotypes are beardless, produce a 1 beard, 1 no beard ratio among the male offspring. What are the genotypes of the parents? So the male would have to be homozygous recessive in order to not have a beard as its phenotype. And the female is going to be heterozygous, because that would make the female non-bearded. But as Nathan's drawing on the board, that would show us that it gives us a 2-to-2 ratio. So for the males, these would both have beards, and both of these would not. So that's a 1-to-1 ratio among the male offspring.
Okay, number 5.
Cats.
So we're talking about the cats, and we're talking about an X-linked inheritance and reciprocal crosses. So it says that recessive B allele is orange, dominant B allele is black, and anything that's heterozygous is tortoise shell color. It says, suppose you cross an orange male cat with a tortoise shell female. It's also important that these are X. So we have a orange male, so it's going to have a little B and a Y, and then a tortoise shell, so it's heterozygous. So it's that. So the cross, this will be the ones we get. We'll get 2 females. One of them will be tortoise shell, and one of them will be orange. And then for the males, since one of them will get the big allele, the dominant allele from the mom, it'll be black, and then the other one will be orange because it'll get the recessive allele from the mom and the Y. So we get 25% of female tortoise, 25% female orange, 25% male orange, 25% male black. For the next one, it says Male tortoiseshell cats are very rare. What is the genotype of male tortoise cat? So we learned that in normal individuals, males have an X and a Y. So normally they would just have a big B or a little b here, but in some rare cases you can get XXY, which will still produce a male, and to get tortoise color you'd have to have a heterozygous X coupled with a Y. For C, it says in a fruit fly, mutation produces a scalloped wing. Assume that the scalloped wing phenotype is a result in X recessive. Indicate the genotypes and phenotypes in the progeny produced when reciprocal crosses are done. So you do a reciprocal cross, kind of like Jordan was talking about earlier, where you take an X, a female that is homozygous for one. So let's just do these again. And then a one that has the trait. So this male would have the trait, and you would get the cross. And then Jordan's going to start drawing out the other one, where you switch, where now the female has the disease and the male does not. And you check the phenotypes and to see how they compare. And if we're able to compare them and see that the phenotypes are the same, then we can say that it is not an X-linked. But as we see, all the males in this cross would not have that because they're going to have a dominant allele. But in this one, they would. So there's going to be difference. So even though the females are the same phenotypes, the males are different. So we can assume that this is an X-linked, which leads in perfectly for the last question because they say to do the exact same thing, but with just an autosomal disease. So you would have one that is big B versus a little b, and you're going to get big B, little b for all of them. And if this is the male and then this is the female, it's the same in both, because then we put the male here and the female here, it's going to create the same. So these would be the same, but since these both aren't the same, we can conclude that it's a X-linked trait. So that's all of them. So sorry about the board. I know it's backwards, but we'll figure it out. Hope this video works and it's a little bit better than us just reading off the answers. Again, like we said in the beginning, Eldon, Kelton, and Joseph are going to submit a video because our schedules weren't working. But hopefully next week we'll have them all figured out so we can do the video together. But all right, have a good week, good conference weekend.